Hello everyone, this is chapter 1 of ninth class Biological Science, Cell, its structure and function. What is a cell? Cell is a basic unit of structural and functional unit of cell, okay, of an organism. Here we have a typical cell. Why we are calling a typical cell? All the organelles shown in a typical plant or animal cell will not exist in every cell. For example, chloroplasts are always shown in a typical plant cell. Yet all the plant cells do not have chloroplast. They are mostly found in the cells of green leaves, tender stems, etc. The organelles and features is most of cells are included in this model. The typical cell provides a way to study cells. Once we arrive at such a model, we can compare any cell with it. Observe the given diagram of a typical plant and animal cell. So let's see the differences between a plant cell and an animal cell. So this is how we draw a plant cell and an animal cell. So what is the differences and what are the similarities? Let's see. A plant cell consists of first the differences between plant cell and animal cell is the cell wall only. Cell wall. Can you make it out here? Okay, this one, cell wall. Okay. And whereas here we don't have a cell wall. Then chloroplast. In plant cell we have chloroplast. But whereas in animal cell we don't have. So let's see first plant cells. What all we have. So nucleus. Nucleus is the basic unit or a membrane of the cell. We call it as a brain of the cell. Nucleus is present. And this is a large vacuole. And beside the or nearer to the nucleus we have endoplasmic reticulum. I will tell you all functions of these organelles. Okay, and this is mitochondria. Just you keep in mind where and what are present in a cell. So what are all the things are present in plant cell? Nucleus, endoplasmic reticulum, mitochondria, Golgi apparatus, chloroplast, cell wall and vacuole and a plasma membrane. See, it is a cell wall and see here they have shown in a white colored way and they have given here. So see here cell wall also in many layers and then again inside we have a plasma membrane. But whereas we here cell wall is absent but plasma membrane is present. Endoplasmic reticulum is also present. Here it is also present. Nucleus is present. Here also present. But see the structure. Okay. It is bound by a nuclear membrane. But whereas here there is no nuclear membrane. Okay. So see the differences also. Mitochondria. Mitochondria is present. Here also present. Cytoplasm. But here we called as protoplast or cytoplasm only. Okay. Centrioles. Centrioles are present here. See the black dots all. But whereas here we don't find. Okay. Centrioles. Golgi apparatus. Plasma membrane. So first in this textbook I will just tell you the activities. What are the activities? How to see the organelles by which by performing what activity. Cell membrane or plasma membrane. What it is? It's a layer. Is a covering of the animal cell. In plant cell there is another layer present over the cell membrane known as cell wall. With the help of the given activity, you will be able to find the cell membrane. How we can find the cell membrane? Just take a Rio leaf. Rio is the plant name. You just tear it and you just put a few drops of salt solution on the membrane and leave it for how many minutes? 5 to 10 minutes by tearing the Rio leaf. And after that what you will observe? And dissolve 1 teaspoon of salt in it, stir it well. So you can see and you can see put the peel in on the slide and cover it with a cover slip and observe it under the microscope. You will observe this pattern. Okay, this is called as sunken stomata and a plasma membrane. Here you can see a plasma membrane. So with this activity we can observe the plasma membrane in a Rio leaf. And another activity for observing nucleus, in which in cheek cells have our cheeks are there, no, you just take a toothpick, not toothpick, or a earbud and just place it under your cheek muscle, just roll it and put it on the slide and put some, I will tell you. So what are the materials you have to use? A methyl blue, just to pour a drop, only one drop on it, just pour a one drop of methyl blue and wait it for a minute and then cover the slide with a cover slip and observe under the microscope. So what you will observe? You will observe this type of structure under the microscope, and you should observe this. You will not observe this type of structure under a normal or a simple microscope or a compound microscope. You have to use the electronic microscope or an electron microscope, then only you can see the this nucleus, cytoplasm and a cell membrane. Okay, keep that also in mind. So what are the parts of the nucleus? Not the cell we are talking, only about the nucleus. In nucleus, in animal nucleus or a human nucleus, what we have? Nucleolus or nucleolus. In nucleus, we have a nuclear membrane. Then inside the nuclear membrane, we have nucleoplasm. Then inside the nucleus, we have nucleolus, and in between the nuclear membrane, we have a pores for the process of this is because for genetic information, for the synthesis of proteins, we need a ports, so that the messages are transferred from through these ports. So nuclear membrane, genetic material, this is a DNA. Okay, and cytoplasm is a fluid. You can just tell that is a fluid part inside the cell, which consists of all the cell organelles inside it, or which all the cell organelles floats inside the cell. So first it was believed that it was a protoplasm, which is a life form, but after the discovery of the nucleus, it was confirmed that nucleus is the life form, or it contains a genetic material, and it is the basic unit of the that it is the main organelle or brain, which is performed by which all the things are performed by. So nucleus, after discovery of nucleus, and then it is again we call it as only cytoplasm only. Cell organelles. What are the cell organelles which are present inside the cytoplasm? There are mitochondria, plastid, ribosomes, endoplasmic reticulum, Golgi apparatus, lysosomes and vacuoles. See, we have seen here in the diagram here. See, these mitochondria. See, cytoplasm. These they have shown like this, but these all things we can only see when we just take its a section. But otherwise we can only see it is in a bean like form, mitochondria. When we open it, how it will be? In that way, if we cut the mitochondria, we can see in what is present inside the endoplasmic reticulum or mitochondria or chloroplast, everything. So endoplasmic reticulum. Let's see first endoplasmic reticulum. So there are two types of endoplasmic reticulum. There are, what are they? Rough and smooth. So why we are calling as rough? Because of the presence of the ribosomes on the endoplasmic reticulum. This is the nucleus. This is the nucleus, and on beside the nucleus we have endoplasmic reticulum, where from ribosomes just carry the information for the process of for the synthesis of proteins, and the protein synthesis takes place with the help of the messenger RNA, and proteins are formed. Okay, and next comes the Golgi bodies. Golgi bodies are also cell organelles which are found in 1898 in a compound microscope you can find. These are thread-like structure which are bulged at the ends. These consists of vesicles which stores the protein. Okay, and they what they will do, their function is to they are mostly present near the cell membrane. What they will do, they will just supply the or transport the proteins where they are needed. So they will store the proteins inside the vesicles. So Golgi complex are very important. They are bulged here at the ends. Lysosomes. Lysosomes is called as the suicidal bags of the cells also. They are the destructive cells, or they produces hydrolytic enzymes which just digest the things which are not needed, or which has to be cut into a small pieces for the process called uh, um, protein synthesis. Okay. Lysosomes destroy almost all the structure in the cell but did not damage it. So it will just destroy it but don't damage it. So what have to be see? Our red blood cell, white blood cells have a particular lifespan. After that they will die. So why they will die? Because new cells have to be developed. In the same way, whose work is over or the lifespan is over, it will just destroy it but won't damage the other organelles. So lysosomes is also called as the suicidal bags of the cell. Lysosomes were discovered as tiny particles visible in cytoplasm. It was found that they contain the destructive enzymes. What is the destructive enzyme? That is hydrolytic enzyme. The enzyme normally do not come in contact with the rest of the cell. The materials that need to be destroyed are transported to the lysosomes. So they are what has to be destroyed. They are transported to the lysosomes, and lysosomes what it will do? It will just produce an enzymes and digest. Means it will engulf it, or it will damage it, or it will destruct it, just break down it. And so that they are called as the suicidal bags of the cell. Mitochondria. Mitochondria is called the powerhouse of the cell. With the help of what experiment or the what activity we can observe the mitochondria? So in an onion peel we can observe the mitochondria, and this is also should be observed under the electron microscope only. Okay, with a normal microscope we can't observe this. So what we have to prepare a fresh solution of Janus green B in a beaker. What do we have to take? We have to take 2 milligrams of Janus green B in 100 ml of water. Take a watch glass and pour some solution. Put the onion peel in the solution and keep it for a half an hour. So first prepare the solution, then put the onion peel into it and leave it for an half an hour, and after that wash it and just put it on the slide, cover with a cover slip, and just see under the microscope. You can see this type of structure. But you can say why we can't you are telling as mitochondria will be like this. Why? But here we can't see like this. We can see only a normal cells like, because this is a just we have opened it. Okay, by just cutting by cutting it vertically, not horizontally, we can see the inside structure of the mitochondria, what are present inside the mitochondria, and what are the functions performed by the mitochondria. They are called, because of this process only, or because of the study of the mitochondria, it is named as the powerhouse of this cell. So what all are present in LS means longitudinal section. Longitudinally it is cut into 2, and inside is like this it is present. Matrix. This is called as matrix. These are cristae. Small you can see here. This will be like a pinballs here on this cristae. This foldings is called as cristae, and on this here we have a small organelles, and in this is the inner membrane, this is the outer membrane. In higher classes we have a huge topic of on mitochondria. You will understand. Okay, ribosomes. What are ribosomes? Ribosomes are where we found ribosomes on the rough endoplasmic reticulum. What they will do? and like structure in the cytoplasm of the cell, they are called ribosomes. They are formed of RNA protein. There are two types. Free ribosomes are scattered in the cytoplasm. Attached ribosomes are on the surface of the rough endoplasmic reticulum. Ribosomes are the sites for protein synthesis. So they are the sites of protein synthesis. So we can found on the rough endoplasmic reticulum also, and on the inside the on floating or scattered onto the cytoplasm. How do we can observe the chloroplast? Chloroplast is a green color like substances. In many plants, or all the plants which are green in color, we can find a chloroplast inside this green color part. So you can take a real leaf again and just stir it, and you can just observe under the microscope. This can be seen with a compound microscope. Okay, and this is the structure of the microscope. This is also cut a transverse section or a longitudinal section. By cutting the chloroplast into two, we can see the inner organelles, which are organ, you can say, inner structure of the chloroplast. That is outer membrane, inner membrane. This is the stroma, and these are called as thylakoids. This is the lumen, and this is the granum. So this is called as the thylakoid. All together called as thylakoid. Each one is called the lumen, and this one called as the granum. Structure of the chloroplast. Vacuole we have seen in the plant cells only. Okay, which is needed for the stiffness of the plant, and also it is needed for the storage of the water, and vacuoles maintain turgor pressure within the cell. They exports unwanted substances from the cell. So what are the key points? Let's see. The fundamental organization unit of the life is the cell. Cells are enclosed by a plasma membrane composed of lipids and proteins. Plasma membrane is a selectively permeable membrane. In plant cells, a cell wall composed of cellulose is located outside the cell membrane. In prokaryotes, nuclear membrane is absent. The endoplasmic reticulum functions both as passageway for intracellular transport and manufacturing surface. Lysosomes are membrane bond sacs filled with digestive enzyme. The Golgi apparatus consists of stacks of membrane bond vesicles that function in the storage. Mitochondria are also known as powerhouse of the cell. Three types of plastids are present in the cell: chloroplast, chromoplast, chloroplast, and leucoplast. Vacuoles are the storage sacs for solids or lipid contents. All cells arise from pre-existing cells. Here I will show you one more thing. These are the keys: C, plasma membrane, selectivity, permeable membrane, prokaryotic cell, eukaryotic cell, chromoplast, leucoplast, vesicles, cristae, matrix. Okay, all the answers I have prepared a video. All the question and answers you can take read it from there, and here is some other by which you can see the overall structure. Okay, cell. What is cell? Cell theory. By whom? Squalden, Schwamm, and Virchow. Cell size, shape, and number. How? These are smooth. Blood cells, nerve cells. How they are? Just you can just give a one. Once you can read it by pausing a video, you will understand what are the functions of which part of the cell. Protoplasm, plasma membrane we have seen, no? So with this you can understand all the functions of the plasma membrane, how well it works. Function of plasma membrane. These are some questions. Cell wall in the plant cell. Okay, function of cell wall. Cytoplasm. What is cytoplasm? Here is another diagram of a cell. Nucleus. Structure of nucleus, function, everything, and it's a prokaryotic cell. How it is present? Single cell organisms, and eukaryotic cell. How it is? What are the organelles present in it? Differences between prokaryotic cell and eukaryotic cell. Cell organelles, endoplasmic reticulum, smooth and rough. Just now we have seen. Function also there here. Golgi complex. See here. This is present near the cell membrane. and we can see these are called as the vesicles, and these foldings is called the cristae, cis phase, lumen, transphase. Okay, lysosomes, the lipid bilayer plasma membrane. These are the hydrolytic enzyme mixture inside the lysosomes, and glycosylated membrane transported proteins. These all we called it as proteins. Okay, see only this small part how it is made up of, they have shown in a magnified it here. Okay, ribosomes, how rough endoplasmic reticulum will be. Function of ribosomes, vacuoles, how vacuoles do what, all that is. Mitochondria, see here, bean shaped. Plastids or a chloroplast, how it will be. Centrosomes we see only in animal cells. Centrosomes, what are centrosomes, how are they? They are in a, they have shown both in a standing and in a vertical and in a horizontal position. See here.